This is episode 98 of the Immunology Podcast, IUIS 2025, Waltzing into the Future of Immunology, with Drs. Wilfried Ellmeier, Laurence Zitvogel, and Dan Littman. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Raud. Welcome back to the Immunology Podcast. We have conversations with immunologists. The Immunology Podcast is brought to you by StemTel Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast could be improved and for suggestions on guests. Today we have Drs. Wilfried Ellmeier, Laurence Zitvogel, and Dan Littman on the podcast to talk about the upcoming IUIS Congress in Vienna, Austria. We also got our usual roundup of recent highlights in immunology news coming up, but first...
Talking about conferences, we are looking forward to seeing you in Honolulu, Hawaii at Immunology 2025. Join us from May 3rd to 7th to connect with the immunology community. We are especially looking forward to Dr. Stephen Jameson's president address titled, I Think You Should Leave, Regulating T-Cell Trafficking and Maintenance. Learn more at immunology2025.aai.org.
All right. Well, I know you've been to Vienna before.
I have. Beautiful city.
Yeah, are you going to book me all the restaurants like I'm about to do for you for Hawaii?
I can try. What should we do? I'm not as big as a foodie as you are. Maybe any of our listeners in Vienna could help us out with some suggestions. But when in Vienna, you're supposed to try the Sachertorte, the Sacher cake. It's this like multi-layered, like very spongy with chocolate layers in between. That's like
Sounds
very
excellent.
typical.
I'm here for that.
Apparently, the cappuccino originally comes from Vienna, or at least some version of it. So,
okay,
coffee.
So coffee. This is happy for me.
Yeah. And also, what else is very typical of Vienna? Subsidized housing. There's something. Vienna is one of the most affordable capital cities in Europe, and I think in the world. So it's also very interesting. What else? I think we should try to go to the opera. Opera of Vienna is really cool. Yeah, we should try to find some ticket. I went once, and I just went to whatever was playing on that day, and it was actually pretty fun. It was an opera, or it was like a theater play of Hansel and Gretel. It was like a children's theater, but that's the only thing on that day. So I figured, well, let's just go. It was really great. So you can feel like in imperial times coming inside the whole place is beautiful. And what else? Yeah, some Habsburg history. It's all over the place. We'll find stuff. We'll find stuff to do. While we prepare for that, let's not forget what we are here for today. Now, what are you going to talk about
I have
today?
a little bit of a blast to the past from IUIS, the last one, where we went to South Africa, because I have South African microbiome data to talk about.
Wow. Okay. All right. I'm interested.
All right. So the two first authors are Dylan Najini and Oyokakere Aduran. I'm sure I butchered that. And the last two authors are Ami Esbahat and Scott Hazelhurst. They came out in Nature the 29th of January, expanding the Human Gut Microbiome Atlas of Africa. So, as you know, there's lots of work going on to study the microbiome, and not just bacteria, but viruses, pathogens of all forms, fungi, across the world to make these atlases so we know what's going on. Well, Africa, as you can probably imagine, is undersampled. And so this paper is really for people in the field who wanted to sample things and to get a better set. And this group sampled both in a variety of places in South Africa, as well as Burkina Faso, Ghana, and Kenya, through some existing networks of sites. And they find a couple of really cool things. So one, they find a decent shift, not just for the region, but also very driven by how rural or industrial urban the place is, with this exception of one of their sites in Kenya, which is relatively urban, but looks like a more rural microbiome. And they have no idea why. So that tells you that there's more going on than just the urban industrial, versus rural mix. But I think what's most important is they found 1,000 new bacterial genomes, and as well as an antibiotic susceptibility factor in one bug, Treponema, and I cannot pronounce as well, sulfafaciens, that might drive its absence in urban populations. But I think the key thing here is they found 1,000 new bacteria that have never been described before, or at least have a genome before, that are quite prevalent there in general. So it's not like, they found this one bug in this one person out of all the people. No, these are like prevalent organisms in their study of what was 1,800 women, because this is also tied to a menstrual study, and so they only use women here. The other thing they found was that HIV infection has a signature that alters the microbiome that was not previously known. So we know HIV in the microbiome. There's HIV infection shifts the microbiome, but they found new bugs that hadn't previously been described before in this paper. So what's the immunology angle and what's everything else? Well, you have a whole bunch of phages and viruses that are influencing the microbiome that have been known before. You have new host commensals, which are really important for sculpting the system. And then you have this tie to HIV that has all not previously been known. And it shows that we, especially as we think about microbiome research, HIV research, and therapy, it shows that our Western biased data for all the underlying data may not actually represent the populations that you're working with, and that you need a broader catch because you're missing a lot of stuff if you try to apply A to B, which is really the point of the paper, in my mind.
That makes a lot of sense, right? I think we see it all across the board in so many studies in which, genetics or microbiome is important. And then, of course, we were only sampling college students in the US or patients in a hospital in Germany. It's going to be fairly biased, and we're missing out on all the diversity that there's across the world. So that's very important for representation, isn't it?
Indeed.
All right. That's very interesting. I have two really cool papers to talk about. So,
You're doing them together again?
no, these are two very distinct topics. So I will explain one at a time. But they're both really nice. I'm really happy. Sometimes, there's some weeks in which I have so much to choose from, but I'm happy with this choice. So the first paper I want to talk is called Identification of Indirect CD4 T cell epitopes associated with transplant rejection provides a target for donor-specific tolerance induction. Whew, that was a mouthful. So first author is Suldig Shanzak. Hope that's correct. And corresponding authors are Christian Larson and Hayden Kissick from Emory University in the US. And this paper is very interesting also because I'm very interested into transplant rejection, and so for me was personally very relevant. But also I think sometimes when we think about transplantation, of course, that rejection of transplants or the generation of immune response against a transplanted organ, it's clearly a immune mediated phenomena. So that's no doubt about that. But I think that oftentimes it's a little bit obscure, like what exactly is being recognized and when, and what are these immune reactions being mounted against exactly? Yes, against the kidney, the allograft, but what exactly in this kidney is that your body sees, or the recipient's body can see, in order to mount, a effective and uh, unfortunate uh, immune response, particularly those more chronic immune responses that involve the generation of donor-specific antibodies. And that's really when you have, a big problem in your hands. So let's talk a bit about transplantation histocompatibility. So in the case of a transplantation, usually you have different stages. Acute, what is known as acute rejection, often happens in the early after the transplantation happens, and this can be accelerated by, the damage that is caused to the tissue during the transplant, and, having to take it from the donor into the recipient. All this generates damage, generates inflammation, and if you have some level of mismatch in the HLA loci between your donor and your recipient, those molecules can be directly recognized by allo-specific T cells in your body, and then you can very quickly reject a transplant. And that was a lot more common before, but now if patients are treated with immunosuppressive drugs, early on, quite aggressive, but then you can keep this acute immune response at bay to some extent, a lot better than we used to. Of course, the disadvantage is that if you really tone down the immune response or the immune system a lot in order to prevent an acute rejection, you are more um, vulnerable to infections, particularly certain viral infections that are very opportunistic. And so it's always, it's a very difficult balance. But then if you survive the initial few weeks, months after the having your new organ, usually, after a year, you can still develop uh, more kind of elaborate immune response against peptides derived from mismatched HLA molecules from the donor. And then you often have antibodies that are recognized in those sequences. So, and that is usually called an indirect recognition, because you're not recognizing the MHC molecules on the surface of the organ itself necessarily, but they are being presented by your own MHC molecules to your immune system, and this allows for really a more complex immune response to be developed, and which includes B cells making, class switch antibodies against these MHC uh, that are mismatched. And why is this important? Because uh, of course, this can end up really in you rejecting the organ. And there's specific combinations. There are certain MHC mismatches that are more correlated with the chance of developing this indirect immune response. And this is the case particularly for HLA-DQ mismatch. It's one of the MHC class 2 MHC molecules. And those have a higher mismatches in these molecules have a higher chance to generate donor-specific antibodies that eventually, create rejection. So what the authors in this paper wanted to understand how is this indirect response being mediated, and what is the relationship with this known phenomena of like there's certain mismatches that are more prone than others. And they use an in silico prediction model, which allow them to, it's called Pierce-2, that scores peptides derived from, now if you have like a mismatch between a donor and a recipient, and then it scores the peptides that can be derived from the donor HLA molecules presented in the HLA molecules of the recipient. And then there's a score on how likely they are to be presented and to be recognized in the context of that MHC. And with this, they kind of looked at a lot of data from transplanted patients, and they show that they can calculate with this algorithm that if they have a higher score of predicted peptides being presented, this correlates with the generation of donor-specific antibodies, and indicating that there is a indirect presentation to CD4 T cells that then, aid B cells to generate antibodies. So that there's really, these peptides are being kind of presented, and this is the source of the antigen to initiate this response. And then you have all these donor-specific antibodies that eventually really it leads to an accelerated organ failure. And so what they do is they look at which are the hotspots that are where these peptides are coming from. They identify there are specific hotspots that have this are much more likely to generate peptides that will be presented in, for example, a DQ molecule has hotspots of peptides that can be presented, or a HLA-DR molecule with high chance. And this really is the thing that has been recognized. So the model is in mice. Mice also have, you can have different strains, will have mismatched MHC molecules, and they can also show that in the situation in which you have a skin transplantation from bulb C to a black 6 mouse, then there are specific alleles that can also, in this case, the H2KD allele in bulb C is different from the H2K allele in mice, which is KB. So B and D, they're different alleles from the same MHC. And this difference is what can serve as an immunogenic target when a bulb C graft is transplanted on a B6 recipient. And this is, again, similar to what you see in the human situation. So what they do is they map the epitopes derived from the bulb C mouse, and they show that they're presenting in black 6 mice. And this stimulates the indirect CD4 T cell response. And basically, these cells, the cells recognizing this indirect antigen, are the ones that are driving the antibody response, the generation of antibodies against these peptides, and the generation of donor-specific antibodies. Even just transplanting the cells will initiate the generation of the adoptive transfer of these cells that are recognizing this peptide is sufficient to induce donor-specific antibody formation, and which really shows that they are instrumental in the rejection, this chronic rejection that models the human situation. And what I think is really interesting is that they provide, and so if you say the recognition of this specific peptides, it's not that many. So that's the interesting thing. It's like really a few peptides seem to be driving the whole antibody response. So if you can prevent the recognition of these few peptides, and you can kind of get rid of the clones recognizing those peptides, that may be sufficient to prevent the antibody response. And they show that if they inject the mice with high dose immunogenic peptides together with a CTLA-4 CTLA-4 antibody, which blockades co-stimulation. So basically binds to CD28 and prevents stimulation through CD28. That can actually prune the TCR repertoire and prevent the CD4 T cell clones that would have recognized that peptide from being activated and initiating the antibody response together with the B cells. And this actually can prolong graft survival, at least in mice, right? This is all in mouse. So basically, by identifying which are the peptides that are being recognized in this mismatch, this MHC mismatch, they show that the CD4 T cells that are recognizing these peptides are driving the antibody response. And if you can get rid of those T cells by stimulating them without co-stimulation, so in a situation in which there's no inflammation and no kind of second signal, that can prune the TCR repertoire in the CD4 cells and lead to lower donor-specific antibody formation, which prolongs the survival of the graft.
Do you think that pruning is fully translatable? So in a human, I could just prune all of it and it will persist, and those epitopes aren't ever going to pop back in the graft because I'm giving them stem cells, right?
No, so this is a kidney. This is a situation of a kidney transplant. So
you'd have to pre-select against that kidney.
So if you have, in this example, they look at HLA-DQ.
Right. But
so if you can identify which are the peptides that are kind of the hotspots of recognition, and if you kind of inject those peptides, so you present those peptides, but not in the context of of an inflammation. Like it would happen if you do a transplant, you have inflammation, you have, you know, all
Yeah.
these extra activated antigen presenting cells that are going to be co-stimulating the T cells. But if you do this before doing the transplant, before there's inflammation, you can kind of, I guess the idea is that they would activate T cells without co-stimulation. We know that in should to some extent result in energy or or kind of cell. You have, once the T cells are activated in this way, they
Yeah,
become
yeah.
energetic.
So I get how you wouldn't want to do it after transplant.
Yeah.
But I doubt that the mice, their system they did experimentally has rip-roaring end-stage kidney disease before a transplant with them on dialysis all the time, which makes the host receiving the kidney transplant not normal and
That is true.
in an inflammatory state to begin with. So I don't know if
Yeah.
we're going to have a clean, quiescent system where you
Yeah.
can go put this in and then prune it through energy, because
Yeah.
end stage renal disease patients aren't, what you call, or I would not call them immunogenic or normal.
Right.
I don't know what they are exactly.
Yeah.
It's a complex question, but I don't know if they're normal.
Yeah. That's a good point. I wonder what, like, kind of like general systemic inflammation, whether that would prevent this combination to work. Um, I don't know, and I don't think they elaborate on that on the paper, but that's a good point. I think that unless like the inflammation is super severe, maybe you can also treat them with some level of immunosuppression before the transplant. Um, I like to think that there should be a way in which you can kind of have the patient stabilize the patient long enough to do this. But yeah, it's probably not as easy as they do in the mice, in which, the mice are already naive and they're just waiting. They're there and they don't have anything severe going on. But I thought it was really cool. So the one thing that they do say is that conventional immunosuppression does not. So they treat them with cyclophosphamide. No, it was calcium channel.
Calcineurin
Cal-
inhibition.
yeah, calcineurin, and that does not work. If they treat the mice with calcineurin and the peptides, that doesn't do. It has to be uh, co-stimulation blockade. So you still need TCR stimulation without co-stimulation. That's the way that this works.
Hmm.
So I think if you do general immunosuppression with calcineurin, for example, something similar, that wouldn't work.
Yeah, I have questions on the practicality in a human versus a clean mouse system for sure.
Well, that's yeah, that's so often the case. I still still think it's really cool because there I've been looking into exactly this kind of antigen recognition in a transplant setting, and I think this is a really clear work showing exactly how do we look at what exact we know that there is indirect recognition, but I like this because you see these are the hotspots of recognition, and it's not that much, it's not infinite, right? It's a very specific part of the HLA that gets presented, and we can to some extent predict it, and I think that's very powerful. I guess you could also say, well, maybe if we could choose one organ, we could try to also use this as a marker, as a biomarker, and say, well, if we know this is a high risk peptide that is highly likely to be presented on the recipient's HLA molecules, we might not want, this might be a bad match because we know that this is a very high matching combination and very likely to stimulate CD4 cells, and we might not want that. Um, so yeah.
It's interesting for sure. Like super interesting. Hopefully it works, but I have concerns. That clean immunology. It's hard to.
Yeah.
All right. Well, I decided to do a COVID paper because it was interesting as a concept, but I have some thoughts on this one. So it's in Nature. It's called SARS-CoV-2 Evolution in the Dynamic Immune Landscape. First authors are N. Alexia Rarininiria. I really killed that. Niles Gubella and Daniela Bornijgen. And the last author is Max von Kleist, and came out on the 29th of January. So this paper does something cool, which is it tries to match real life data with a model that can predict evolution of COVID over time. And so it goes, look, if waves of COVID, as it mutates, should be associated with neutralizing antibodies not being useful anymore for COVID, right? As the virus mutates, your neutralizing antibodies you have stop working, then you get sick again, which is why the vaccines don't prevent transmission. They just prevent us getting really sick because neutralizing antibodies don't last long. So they took the wave information alongside the sequencing of the spike protein, because that's kind of how you identify all these different mutations that stop the neutralizing antibody. And they said, hey, can we predict what actually happened just by the mutational sequence? So can we say, hey, once you have this much drift from something that previously hit, you'll get a pandemic, you'll get another wave or not, essentially. And so what they were able to do is they looked at historical variant dynamics, and they were able to separately predict in their model future variant dynamics, especially in terms of evolutionary pressure, right? Because if the entire environment is immune to that one, it's going to shift away from that. So they're able to predict the dynamics and the population waves over time of what's happening. So they could say, oh man, this variant came in, it's going to smack everyone, it hit everyone. So now it has to go this way, towards these types of mutations to then have another spike. And that's what happens. So on the one hand, it's super cool in that it shows the kind of what we think of evolutionary theory and what's happening works. And you could use this to predict where mutations are likely to go to build future vaccines. But it also seems like there's enough different options that you're not going to predict it right. You'd have to have like a multiplexed RNA vaccine. And I don't know if there's a point, right? The vaccines that we have, regardless of the specific variant, protect us from severe disease. Respiratory viruses that mutate at the frequency that COVID does, and most respiratory viruses, are almost impossible to vaccinate against to get herd immunity from preventing transmission. They just mutate too fast. And this paper essentially shows they mutate this fast, but we already know they mutate this fast just from the real world data. So on the one hand, this model is really cool. From a genomic surveillance perspective, it maps like evolutionary theory and genomic prevalence to what really happens. And that's neat to see the theory and reality match up. So I really like it from that perspective. Clinically, I don't know if it gets us anywhere. I don't know that we could go use this, predict a mutation and get it out so much faster and then be right often enough to have any benefit. And it's just going to mutate again in the meantime. And unless you're pumping out vaccine weekly with new variants and pumping it into people, you're never going to get ahead of the virus enough to stop it because it's now just endemic. And so that's where I'm kind of like mixed on the paper. But very cool, like that you can see that, oh man, everyone here had this, or they'll also see like, hey, this site got this huge variant, looks like this. So when this other wave comes around, places who's already were previously exposed to something very similar to the new wave had way lower incidences of a new spike. And so they could really see that the mutational drift was predicting recurrence. Like you have to have enough drift away from what you previously had to get a new wave. And it worked in all directions as different things circulate the world. So that is really cool.
And so is this also applicable to other kind of viruses that kind of change, like, I don't know, like flu or things like that?
Theoretically, flu is weird because of it has a different binding epitope system and recombination. right? It's not, it's, the H1N1 or whatever you call it. Those H's and N's are different parts of the virus that recombine and then also mutate. So the dynamics are much more different than how much drift in the spike protein of the virus do I have. They've been trying to predict flu for a while, and we suck at it.
Yeah. Yeah. I mean, we try, right? We try to see which is
We're terrible.
which is next year's strain is going to be but
we're like less than 50% accurate.
Damn. Oh, we try.
So this is why I'm like, this is cool, but also, is it useful?
Right. Yeah. Yeah, especially if making vaccines, it's well, we kind of did it once, but it's a lot of work to just, even if we have the information, whether we can actually translate that into making vaccines last minute based on our predictions is tough.
Yeah.
All right. Okay, so to finish our conversation today, my second paper, also really cool. And this paper is titled "The Meninges Host a Distinct Compartment of Regulatory T cells that Preserves Brain Homeostasis." The first author is Miguel Marin Rodero from the lab of Diane Mathis at Harvard Medical School. And I think I have a soft spot for Diane Mathis lab work, because I think it's like the third paper I present already, because they always do such cool things, specifically with T-Rex in different places. They had really cool work on T-Rex in the muscle and how important they are to keep the health of the muscle and to respond to exercise and increase, the our increase of performance after exercise depends on T-Rex keeping the immune inflammation at bay to allow for the muscle to recover. And unsurprisingly, T-Rex have a similar role across the body. And so in this particular case, and this is a story that Diane Mathis had already presented, I think it was at AI, so I was a little bit familiar with it. Let's talk about T-Rex, not necessarily in the brain, but in the meninges. And the meninges are this layer structure that kind of envelops the brain and the spinal cord. And it has become evident lately that they are an active immune environment. It's not just like these three membranes around the brain keeping the brain fluid inside, but they're actually a lot more dynamic than that. And we know that there are immune cells in there, and including macrophages, the dendritic cells and lymphocytes. So there's definitely lymphocytes there. And we already know that adaptive immune cells influence the function of the brain to some extent. Like the presence of immune cells is important for normal cognitive function, behavior, and I find that super, super weird, super cool. And I think this paper goes away into kind of our understanding of why. So regulatory T cells, I think I've spoken about many, many times about them. They we're going to be talking about CD4 T cells that express FOXP3, and they can maintain immune tolerance. They can inhibit other immune cells, and they do it through various mechanisms. But one of the mechanisms that are really important for this paper is their capacity to consume IL-2 with their high affinity receptor. CD25 is the alpha subunit of the IL-2 receptor, and they express a lot of it. And this is very important. They also make anti-inflammatory cytokines, and they can also interact with antigen-presenting cells, and modulate their function. So they do a lot of things. And so what they do in this paper, which I can imagine must have been a lot of work, because you are looking at the brain of these mice. You have to like dissect the brains and take, the different layers of meninges apart, and they end up with very few cells. Like they estimate like literally hundreds of T-Rex per mouse in their brains. That's very few. But when they look into the meninges of the mice, they do flow cytometry analysis, and they use FOXP3 reporter mice that express FOXP3, GFP, and they show that there is indeed a distinct population of FOXP3 positive CD4 Tregs in the dura matter of the meninges, the outer layer, and that is in higher abundance compared to other parts of the meninges. What is interesting is that these Tregs are clustered alongside MHC class 2 antigen presenting cells, and they show some microscopy data, and they show that they stain for MHC2 and they stain for FOXP3, and they see FOXP3, GFP, and they see that they're kind of clustering next to them. And these are probably kind of dendritic cells and Tregs. Meningeal Tregs increase with age of the mice, and this is also the case for other tissue resident Treg populations. So they show that within this population of meningeal Tregs, which, as I said, is very small. So let me just double check, they find about 125 Tregs per mouse of meningeal Tregs. It's very small. So you see the cytometry data, and you see that sometimes they show the dot plots, and it is a little bit of a matter of faith sometimes because it's very few events. But I can imagine this must have been really hard to do. So I like that they have also microscopy data, because that is a little bit more convincing sometimes. And there's different types of Tregs. They kind of do single cell RNA-seq, and they show that there's a subset of Tregs that are expressing TBET and CXCR3, and these are Th1-like Tregs. There's some follicular regulatory Tregs, and I think most interesting, this is Th1-like Tregs subset, because they are good at regulating interferon gamma responses. And in the end, this is kind of the most interesting finding they have, is that if they deplete Tregs from this, and on this population, they treat mice that have a diphtheria toxin receptor expressed specifically on Tregs, and then they treat the mice with diphtheria toxin. This is a very commonly used mechanism to deplete specific subsets of cells. And they show that this deletion of Tregs, this is in a short time, so kind of they try to avoid a systemic effect. They are really focusing on the brain. And they show that this results in a rapid increase of interferon gamma producing CD4 T cells and CD8 T cells in the meninges. And no, this is, they look into this, and it really seems to be a consequence of the absence of Tregs. Then these populations, they come and they infiltrate, and they generate interferon gamma mediated inflammation. And it's not super clear where these cells are coming from, so they don't really explain that, but they do show that they increase in their numbers in the meninges, and they produce interferon gamma. And the thing is that interferon gamma is very detrimental to neurons, and the loss of Tregs induce microglial and astrocyte activation, and this kind of generates this neuroinflammatory state, and this is bad, particularly in a place in which we have like the one part of the brain which has the kind of the highest plasticity, and where you still have neurons and connections being produced a lot, which is the hippocampus, where which is related to memory formation. And particularly in the hippocampus, and different structures within the hippocampus that I don't think, I'm not going to explain much, but this interferon gamma really affected the proper maturation of the neurons in the hippocampus, and eventually results in deficits in short-term spatial memory. They have this MACE assays, and which they look into the memory of mice, and this results in both markers of inflammation in the neurons and reduced memory formation in mice. And what they show is that the way in which Tregs are preventing this interferon gamma production is through the consumption of IL-2 in the niche of the meninges. So if they neutralize IL-2, then you don't see the effect of the absence of Tregs is kind of gone. So there's no so much interferon gamma production or immune cell expansion, T cell expansion, if IL-2 is not there. So really looks like, and this is something that I know has been described in other systems, that having the Tregs there consuming the IL-2 quickly prevents the activation of STAT5 and down. So the STAT5 activation in conventional cells, which results in less production of interferon gamma, which protects the neurons, and in the long term, safeguards brain homeostasis and cognitive performance of these mice. And I think there's other. So this is really cool. So basically, inflammation in the brain bad. Tregs, resident Tregs keep inflammation at bay. Particular interferon gamma, which was also the bad actor in the case of muscle inflammation. Also, interferon gamma was bad. It's very interesting to see how detrimental, how much the effort we put into keeping interferon gamma at bay in sensitive tissues.
Well, once it starts going, it really starts going, right? And it brings everyone to the party.
Exactly. Yeah, it's such a powerful cytokine, interferon gamma. It's so important to keep it at bay.
Well, this is interesting. I want to believe that they found 125 Tregs in a mouse. But
yeah.
They found 125 Tregs in a mouse.
I mean, the description of the method is just, they're just like,
I'm sure they had to cross-confirm it. And
I mean, I think the microscopy data, there's a bunch of microscopy data and they really show that you can see them inside. Okay. So I think that's really helpful. That really adds to the what's the word? To the
faith.
Yes. Reliability of these results.
Well, in addition to powerful cytokines, we're going to be speaking about a powerful scientific meeting with this IUIS Congress coming up in a moment. But before we get to that, this is time to remind you to register for that powerful meeting. Early registration is open until April 15th for the 19th International Congress of Immunology taking place in Vienna, Austria from August 17th to 22nd. Register now at www.iuis2025.org.
For the second part of today's episode, we have a very interesting conversation. We have actually 3 guests joining us today because we're going to be talking about the upcoming IUIS Congress that is taking place this year, 2025, in Vienna, in Austria, between August 17th and August 22nd. So this will be the 19th edition of the International Congress of Immunology from the International Union of Immunological Societies, IUIS. And we have with us 3 guests that are, going to be organizing and speaking at this conference. So we're very excited. So first of all, we have Wilfried Ellmeier. He is professor of immunobiology and head of the Institute of Immunology at the Medical University of Vienna. So he's going to be acting as kind of our host for the Congress. He's co-chair of the Scientific Planning Committee at IUIS. We also have Laurence Zitvogel, who is professor and group leader at the Institute Gustave Roussy that's in Paris, France. And we have finally Professor Dan Littman, who is at the Department of Cell Biology at the NYU New York University in Langone in New York. And so both Laurence and Dan are going to be giving keynote talks. We're very excited to attend and to enjoy the conference this August. So welcome, everyone. Thank you so much for joining us to talk about the upcoming Congress and a little bit about your research as well.
Thank you all for being here.
So maybe I start as a host. So welcome also from my side, from the Austrian side. My name is Wilfried Ellmeier, and together with my colleagues Rodolf Valenta and Barbara Bohle, we are responsible as local hosts for organizing the 19th International Congress of Immunology, IUIS 2025 in Vienna. We are really happy and excited that in a few months from now, we will be able to welcome many international scientists in Vienna. We expect approximately, hopefully 4,000 people, maybe a little bit more, from all over the world, so that actually Vienna will serve in late August as a melting point for immunological research worldwide. We have put together, hopefully, an exciting program with keynote lectures, 2 of our keynote speakers together here with us. We have plenary sessions. We have planned a variety of different symposia. We have planned meetings, symposia of the IUIS committees, but also symposia based on proposals from our scientific program committee, from national societies. We will have sessions about career development, so for our young colleagues, and also we will have special sessions about meet the editors. And of course, there will be also a great social program, but I guess I will talk about this a little bit later in our podcast.
Maybe we can start asking our keynote speakers with us today. Maybe ladies first, Dr. Laurence Zitvogel. Would you like to introduce yourself a little bit for our listeners and maybe give us a sneak peek of what you're going to be sharing with us in August? I know it's a long time to go, but you have to prepare for this in advance.
Yes, so I am an oncologist trained at the University of Paris, University Paris-Saclay. And originally, I was an immunologist by nature, and I became a microbiologist by opportunities. In fact, onco-immunomicrobiology is a nascent. It's a nascent field. So I am more interested in sensitizing and raise the awareness of what is gut dysbiosis. So why perturbation of the gut microbiome repertoire is a problem or a disease or a risk factor in oncology. So I'm going to define what is gut dysbiosis, at least in oncology, based on metagenomics and some MATCAM-based mechanism as a first definition. Then I will discuss the causes of gut dysbiosis. For instance, the importance of comorbidities, co-medications, antibiotics, proton pump inhibitors, benzodiazepines, for instance, and also how oncogenesis per se is actually causing gut dysbiosis. Then we will allude to consequences of gut dysbiosis at the level of the systemic immune tonus and the cancer immunosurveillance, at the level of metabolism. And I will then try to tell how vaccination can be compromised by gut dysbiosis, and how the success of immunomodulation by immune checkpoint inhibitors and CAR T cells can be hampered by gut dysbiosis. Finally, I will discuss the therapies and why we should go for fecal microbial transplantation or live biotherapeutics or some prebiotics and diet interventions. And I would like at the end to raise awareness of the importance of looking and investigating for mode of actions and creating some kind of clinical biome in oncology department all over the world. So meaning that we should incorporate at each time a patient should be managed. We should try to include some investigations at the level of the gut dysbiosis. So finally, we have to discuss the clinical and the biological relevance of bacteria that are invading cancer beds. So this is an evolving field. Oncologists are a little bit afraid of adding this component to all the other layers that are involved in the understanding of why a patient is doomed to fail therapy. So I understand that it's maybe the complexity is refraining a lot of minds from getting inside this field, but I would like to encourage young investigators to submit abstract, to join the session, to discuss with us and see how joint effort throughout ethical differences, geographical differences, and so on can be overcome by joint efforts.
Yeah. I remember being so surprised when that work from your lab looking into the relationship between the microbiome and response to checkpoint inhibitor was published. I thought it was exactly like not yet another layer of complexity to this. It's like, how are we ever going to get through to this. So I'm very much looking forward to hearing more about that particular topic that for me is just mind blowing.
Well, it's what I do for a living, right?
And Jason just lives for that, right? That is right up your alley, isn't it?
I'm the head medical and scientific officer for VOWST at Nestle, so we may want to talk later.
Yeah. Yeah.
I think we have one more, right? Coming up.
Yes.
I think, Dan, do you want to give your kind of 2 minutes?
Sure. Yeah. Well, thanks very much for having me participate in this. I am trained as an immunologist and also a physician, although I've been doing research my entire career. And we've had a very long-standing interest in understanding how T lymphocytes develop, initially being trained in the thymus. But more recently, we've come closer to the sorts of topics that we heard about from Laurence, trying to understand how exposure to the environment during the lifetime can influence the immune balance, particularly from the T cell perspective. And that means mostly through exposure to both diet stimuli or antigens and commensal microbiota, the microbiota that we live with in a mutualistic way. What we have now recognized is that T lymphocytes throughout lifetime are constantly being stimulated by antigens from the intestine in different parts of the intestine, for example, in the upper part of the intestine by food antigens, and then throughout the mostly lower part of the intestine by microbiota. And that this is critical for maintaining health throughout the body. Laurence is very focused on cancer and on anti-tumor responses influenced by the microbiota. But in general, these kinds of stimuli that are encountered in the gut will influence processes throughout the entire body and can contribute when they are dysregulated to autoimmune diseases, as an example, and certainly to allergies, the different types of allergies, asthma, as well as, of course, food allergies. So our focus and what I'll be talking about is to try to identify basically the intermediary cells that connect the molecules or the antigens that are seen in the intestine to the T lymphocytes. And what we've recognized over the last few years is that each different type of T cell seems to be instructed by a different kind of intermediary antigen-presenting cell. So that's what I'll be talking about, and in particular, whether it may be possible to harness some of these antigen-presenting cells, particularly the ones that can promote tolerance. And this is the opposite of what one tries to do with cancer immunotherapy. In this case, what we'd want to do is to target these cells to be able to induce tolerance to sometimes to self-antigen, sometimes to food antigen, depending on whether people are suffering from allergies or from various autoimmune manifestations. So I think it's a very exciting area that we're currently studying, and I particularly look forward to this meeting because it brings together people from all over the world who often are not exposed to the kinds of things that some of us are very fortunate to be exposed to quite regularly, and we hope to be able to really broaden the appeal and the opportunities for such people to participate.
I just want to say for long-time listeners of our show that we did discuss your publication. I mean, it's been already some time about the ROR gamma T positive dendritic cells and the induction of tolerance. So I hope that our listeners are, maybe they should check out that episode again before attending to your keynote. But that's very exciting. I thought that the idea of having, really specialized antigen presenting cells to induce this, in this case, regulatory T cells and tolerance, and how important their existence is for proper development of tolerance. it was really, really cool. I really enjoyed that, discussing that particular publication. And so we're looking forward to hearing more about kind of how the story kind of expands or for other cell types as well, or other situations. So very exciting.
I hope to have more to say and to sort of highlight the opportunities for therapeutics there.
Yeah, for sure. Right. That's really often the question in our minds. How do we harness this for therapy?
Yep.
And I think what would be in common between Dan and my work is the importance of biliary salts, biliary acids. So there are many of them. They are, they have been underestimated in other field aside from steatosis and so on. So digestive tract related diseases, but in fact they have broad bioactivities on different components of the immune system. And I think that this deserves some highlights from Dan and I in different settings and context dependencies. So I think this is also an underestimated field that needs some more interest from the audience.
For sure. Very, very exciting.
So it sounds like I'm going to have fun at IUIS this year.
Absolutely.
Why should people in general, our audience listeners, want to get on a plane and go to Vienna? Starting with the conference, we'll also talk about Vienna because both are important. But starting with the conference, why should people go fly out to see you guys?
I think Dan mentioned it already a little bit, because it's kind of the one conference which is really dedicated that all people across all over different regions from the world meet in one place. Because very often you have conferences, smaller conferences, which are dedicated to a particular topic, to a particular field. And of course, then the community comes together and talks about it. But having a kind of an exposure to a variety of different fields and being able to meet really the top leaders in the field and coming maybe from countries that are less economically rich and with possibilities than maybe from the more developed world, as Dan just mentioned. I think that's one of the key issues, what makes IUIS so special in terms that it brings together the people, immunology without borders, and that actually then in Vienna is the unique opportunity. I think one of the reasons why people should also come to Vienna is it's a central city, somehow easy, not only in Europe, but it's easy to reach from everywhere in the world. I think it's good to reach, it's easy to reach. It's also affordable city. It's not an expensive city, especially for students coming from low and middle income countries. There's very affordable hotel accommodations and pensions in Vienna where you can stay. So you can stay probably for 60, 70, 80 euros in a very nice place. Vienna is a safe city. This is good. So we also tried IUIS conference here that we provide travel fellowships for students and postdocs from low and middle income countries. There's also a special registration fee which is reduced in comparison. So we really try hard so that we really get a lot of people from all over the world to Vienna. So this is probably one of really the unique selling point, if you want to say in that way a little bit, of why an IUIS Congress is so special in comparison to many other conferences. It's really, and it's broad. And as already mentioned, I think one thing is also important that you really connect basic research, then also with translation and then perhaps the clinical application, that you really meet people and from all of the different directions of this kind of translational value chain. And we have talks about basic research, translational, we have sessions, symposia, bringing rather the clinical aspects of the immune system into attention. And I think the combination of this makes it unique, and really hope that people will come. In addition, Vienna is a great city. I really enjoy living there. I think people are really spoiled if you live there because it's really such a nice city. I think even in the world ranking, it has, I think, a city with one of the most highest quality of life, I think, in the world. What's also one interesting aspect is the cultural aspects. I think I don't want to advertise now. I think there's classical music. Actually, 2025 is a special year because it's the 200th birthday of Johann Strauss. It's one of the king of waltz. I think some of you might know, on the Blue Danube, which is a kind of a very famous waltz. And therefore we also have as a motto that actually the motto for IUIS is waltzing into the future of immunology for a better health. So this is, maybe that was the reason why the Congress was then also awarded to Vienna. And I think it's really a nice city. You can experience the city. The Congress venue is close to the city center, so it's easy to commute from the city center by subway. It's less than 10 minutes. You're at the conference center and back. It's a pleasant city. We also are planning a very nice and great social program. Of course, there will be a welcome reception. We will also have a great Congress party. We're also planning to have an immunology run, where we try to collect money to provide more travel fellowships. So this is also something what we will do. And by doing this, by having the kind of special scientific program also embedded in a nice city environment, in an affordable city environment, where you have cheap possibilities to reach the city, to stay in the city during the conference time. So we hope that we can attract many people attending the meeting. And of course, as an example here, we have great science. Please also visit the website. We are constantly filling up the program. It's at IUIS2025.org. We have great keynote speakers. So in addition to Laurence and Dan, we have Yasmin Belkaid, Akiko Iwasaki, Ruslan Metzidov, Andreas Radbruch, Shannon Turley, Shimon Sakaguchi, and Rudolf Valenta. So I think covering a variety of different topics. So I think it's really worth coming. The plenary sessions we have already, the list of speakers. Now we are filling the symposia. And as already mentioned before, we will have additional sessions. So to really also foster interaction, career development, helping young students to get their papers published in the top journals. So we will have, as I've already mentioned before, meet the editor sessions. So there are many things, and I think it will be a lively conference.
Yeah, we have to say that it's a love story between the immunologists, because the key word is immunology. You know, when you love immunology, you love it, at the exponentially. And seeing that this is a unique place, a unique opportunity for immunologists, young ones, senior, the ones that are really infatuated by this discipline, to meet across different fields, whether you talk about autoimmunity, cancer, infectious diseases, allergy, and so on. You can cover the knowledge and meet the experts in a really private way. And it's really a unique opportunity for everyone to really enjoy and practice their immunology.
To reemphasize, how important it is to take advantage for people who have the access to take advantage of these outreach programs to have funds to attend the Congress. As Wilfried mentioned, it's obviously really important for those from lower income countries where we need to do everything we can to provide more of such funds. But I should also point out that even in the United States, the American Association of Immunologists provides special funds for this kind of travel to the IUIS Congress. And what I've seen to my dismay in the last couple of congresses is that very little of the available money was actually accessed. And so I would just urge people to take advantage of this, because the money is there for attending this Congress. And there needs to be maybe more awareness of this.
I fully agree. And maybe one additional point, I think it's also a chance for students, if they join, I think you can meet perhaps maybe your next lab leader, right? I think you go there, you can talk with them, you can directly interact face-to-face with them. We will provide enough space and time for poster sessions. There will be also abstracts selected for oral presentations, but we extended the poster presentation time. So there's a lot of time for discussions. And if you are a young student and want to join Dan's lab or Laurence's lab, just go to them, tell them you have a poster, please visit my poster. That's what I've been doing during my PhD. And these are unique opportunities. And I think this is really something, it's much better than writing an email, right? just and sending a CV, but if you have the possibility to interact physically with them live, one-to-one in Vienna, I think these are the opportunities you should take advantage of.
Yes, I wanted to make that point exactly, that especially if you're coming from, a more peripheral country, I'm from Argentina, so it can be really precious to have access to people from all over the world, from labs from all over the world, and especially if you are, reaching a transition, if you're finishing your PhD, if you're, looking for a new postdoc position, this is great. People like meeting people face to face, having the chance to kind of make your case in person, especially if you bring a poster that you can, talk about, that your research, that is so powerful. And so also from my perspective, especially those people coming from, from Latin America, from Africa, from Asia, this is an opportunity in which you could get, a stipend to attend. For what I understand, the sponsorship includes, accommodation. You can get the fee waive, and have all the opportunity to attend and meet everyone. So this is something that people definitely should check and submit an abstract, ask for, and try to be considered for a scholarship, and go for it, right? And then make a list of people you want to meet and try to find yourself in their presence at some point. You have a few days for that.
I was going to ask, so why to go to the conference? General, high level. We've heard a couple of keynotes. Are there any big themes of this conference this year, or kind of like organizing principles across the field of immunology? We heard a little bit about the microbiome thing, which makes me a happy little camper. But anything else that are going to be big themes that we should, as guests, anticipate wanting to listen to, or big, pillars?
Well, I mean, in terms of pillars, I think, if one organizes a conference, one doesn't have to reinvent the wheel, right? I think somehow you have a little bit of kind of a track-like structure. But again, we really try to cover all the most important topics. I think just for the plenaries, and I think we will. I don't want to list now, but it's about allergy, autoimmunity, also artificial intelligence and systems immunology. I think that's important topics which are here already, right? It's not that they're coming. I mean, they're already implemented in our daily life probably, but also in our research life. So this is something. I mean, cancer immunity, immunotherapy, of course, is a big topic, but CAR T cells, but it's also exciting developments that CAR T cells are now used also to combat autoimmune diseases, right? So I think this is something we would like to cover, and not to mention innate immunity, inflammation, infectious diseases. Also one emerging topic is neuroimmunology, so the link between the neurosystem and immune system, development of vaccines and novel approaches. So I think these are all the topics which are covered. And of course, so this is some topics for the plenaries, and then we are more granular on the symposia topics. I think immunity across all ages are B-cell immunity. Then you have also cytokine regulation of immune. I mean, I could list now a lot of different topics, but really, please visit our website. But we really try to be broad. And then for the oral abstract sessions, we even have more topics, right? They're all listed on the website, and we really try. But there are exciting recent developments, and we try to cover them. And again, coming back, what's so special about the IUIS 2025 Congress, or IUIS, is that it brings together a lot of different topics. Because if I'm working on T cells, so epigenetic regulation, if I go to a meeting, then it's very focused on a particular topic. And I will never hear about something, how CAR T cells are used to treat autoimmune diseases, right? But actually, coming now to a big congress, there you have the possibility to also get exposed to this. Or T-cell exhaustion, infection, cancer, right? This is a fascinating topic. For instance, I mean, there are many fascinating topics, but this is one where you really see how many different subsets exist, and that the response different to immune checkpoint blockade. So I think this is really quite exciting, and we really try to bring from different topics really the latest developments, so that somehow that you feel after leaving the conference, you have now read, I don't know, all the most important review journals from the last 5 years, and that you get kind of a good foundation. So when you go back to your own work, that you can maybe connect your own data with different things you were not thinking about before, or when you discuss at the posters with your colleagues. And maybe one more point, I think one should not underestimate also, you make friends for life when you go to a conference. I remember when I went to my first conference, it was a Keystone meeting in the US as a postdoc. I met people there, were postdoctoral fellows from other labs, and I'm still in contact a few years later. Let's put it this way. So I think this is something which is really nice. You can form your network, and you need your network, and you can connect with many people. So I think in addition to all the scientific aspects and the input to your own work, I think it is also a big impact on to form your own network, particularly if you're a young scientist, to connect with people all over the world. So that's also a great aspect.
Yes, I guess we will also hear about the human and mouse immune cell atlas, the new cell types, throughout this single cell analysis and so on. And we also learned about new models, mouse models, new knockouts, conditioned knockouts. So, it broadened the opportunities for young individuals to really dig into the mechanism of action of their own work. So this is also very useful to have access to this late-breaking news, late-breaking tools. So unique opportunities once again for the chauvinist in immunology.
Yeah, in that light that Laurence mentioned, in the past, some of the most exciting advances were reported at the International Congress. I remember when I was still a postdoctoral fellow, I wasn't there, unfortunately, but I remember that the first description of the cloning of the T-cell receptor was at the International Congress in Japan, and it made its way across the world like a wave, like a tsunami in a way that for those of us interested in T-cells, that was quite a monumental event. So hopefully something like that will occur at this Congress as well.
Would be great. So we are all eager for that. Yes.
Sounds very exciting. All right. So I think we're reaching the end of our conversation. I just want to say that also the Immunology Podcast will be having a live show on Thursday, August 21st, so the day before last. So we are working on that too. So that's another event happening. We're very excited. So sorry for the shameless advertising here. But with that being said, I thank everyone for joining us today to discuss this. Thank you so much. We had Professor Wilfried Ellmeier, Dr. Laurence Zitvogel, and Professor Laurence Zitvogel and Professor Dan Littman joining us to talk about the upcoming IUIS conference in Vienna from August 17th to 22nd. The abstracts submission is open until the day of the release of this episode, so February 11th. But the early bird registration is open until April 15th. So if you want to come without presenting, but late abstracts can be presented later on. And I guess maybe if there's any specific requirement, will be for poster presentation. There will be a different late abstract deadline presented, but for like applying for financial assistance or things, you will need to present your abstract until February 11th. So we'll try to advertise this before the release of this episode. So thank you so much for joining us again to the Immunology Podcast, and we're so excited. We're so excited to go to Vienna this August. Jason is so excited to come to Europe, to the old continent. I live in Amsterdam, so for me, I hope to be able to take the train. We'll see if I can make that work.
Yes.
Yes, so take an overnight train. That will be a sleeper train. That will be fun. But Jason, you'll have to fly. Sorry.
It's a tough life. I'll manage. Well, thank you for coming on.
Hey, thank you very much for inviting us, and we are also very excited to meet everyone there.
Yeah, also thanks for having me, and I'm looking forward to seeing all listeners live in Vienna, August 17th till 22nd. Yes.
Great. Look forward to seeing you all.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and roundup papers. You can also reach out to us on X at @ImmunoPodcast or via email at info@immunologypodcast.com with feedbacks or just guests. See you next time.